Hi, my name is Adedoye Victoria Aluko. I'm a master's student of biotechnology and chemical science. This is my last term here in Roosevelt University.
Hi, my name is Damilola, Sunmola, a master's student of biotechnology and chemical science management, and we are here to talk about the exploration of novel imidazo 1,2-alpha pyridine-3-carboxamide derivative as a potential antimicrobial agent, and we have Dr. Onajole, who is the principal investigator. So this is just the content of our presentation on what we're talking about today.
Thank you. Mycobacterium tuberculosis is a rod-shaped pathogenic bacterium, which is the major causative agent of tuberculosis. This disease infects the respiratory organs, especially the lungs. About 1/4 of the world's population actually has latent TB. That means they've been infected with Mycobacterium tuberculosis, but they do not show symptoms. TB has been one of the major causes of death, and it has killed about 1.5 million people in 2018. The 30 highest TB burden countries accounted for 87% of new TB cases in 2018, with India leading the count, followed by China, Indonesia, the Philippines, Pakistan, Nigeria, Bangladesh, and South Africa. This is according to WHO. Some of the virulence factors are listed here. We have mycolic acid, slow generation time, presence of cell wall lipids such as PGIM and PGL. These virulence factors have made it resist medical treatment, hence it becomes multi-drug resistant. Multi-drug resistant TB is a case whereby TB is highly resistant to the two frontline medications, which are rifampicin and isoniazid. It remains a public health crisis and a health security threat. Therefore, new drug regimens are urgently required to combat this deadly disease. Here is a picture of the scanning electron micrograph of Mycobacterium tuberculosis. A lot of drug discovery programs have led to the discovery of promising anti-TB drug candidates. We have drug candidates such as BM212, which is a pyrrole derivative, SQ109, which is an ethylene diamine derivative, AU1235, which is a urea derivative, and Q203, which is an imidazo 1,2-alpha pyridine derivative. These are awesome anti-TB drug candidates because they are readily synthesized compounds. They have been found to be ideally suited to structure-activity relationship, which has to do with MIC values. They have low molecular weight and they have ideal log P values. They have good metabolic stability and they have tunable pharmacokinetics. So members of the research team to which we belong to recently discovered two imidazo 1,2-alpha pyridine derivatives with excellent anti-TB activity against susceptible and drug-resistant strains of TB. Our main interest here is actually to look at these drug candidates, especially the imidazo 1,2-alpha pyridine derivative, and also to find out if they not only contain antimicrobial properties, but they also contain antibacterial properties against the selected organism. Here's a structure of the Q203, which is an imidazo 1,2-alpha pyridine derivative that we'll be talking about today.
So for the experimental objective, the aim of this experiment is to investigate the antimicrobial properties of OK188 and MY09 against 8 bacterial microorganisms using broth microdilution method. So for gram-positive bacteria, Staphylococcus epidermidis is a gram-positive coagulase-negative cocci that is part of our normal flora. It is the species found most commonly as a member of the normal flora of the nasal mucosa and the obligus of the newborn. So we have Staphylococcus aureus. It is often positive for catalase and nitrate reduction and the facultative anaerobe that can grow without the need for oxygen. Staph aureus, along with similar species, can colonize and act symbiotically. You have Enterococcus faecalis, normally lives harmlessly in your intestine. However, if it spreads to other parts of your body, it can cause a more serious infection. Then we have Bacillus megaterium. It's a rod-like many aerobic spore-forming bacteria found in wildly diverse habitats. Due to the resistance of their endospores to environmental stress, as well as their long-term survival under adverse conditions, most aerobic spore formers can be isolated from a wide variety of sources. So these are pictures of the 18 to 24 hours cultured plates. Here we use the streaking method, which is a popular method for isolating specific bacteria from a sample containing a mixture of microorganisms.
Just like my partner talked about the gram-positive bacteria used in this experiment, I'm talking about the gram-negative bacteria used in this experiment. We have Enterobacter aerogenes, Klebsiella pneumoniae, Pseudomonas aeruginosa, and Salmonella typhi. Enterobacter aerogenes, Klebsiella pneumoniae, and Pseudomonas aeruginosa are rod-shaped bacteria. They are the major causative agents of infections such as gastrointestinal infections, HAIs, septicemia, UTIs. HAIs are known as hospital-acquired infections. UTI are known as urinary tract infections, adult meningitis, amongst others, and Salmonella typhi is a facultative anaerobe that majorly causes systemic infections. All these organisms were carefully selected because of their biological importance and because they are major causative agents of infections. And the above organisms have become highly resistant to several classes of drugs, making them multi-drug resistant. Here's a picture of the culture plates we used. We have E. aerogenes, S. typhi, and K. pneumoniae. They were carefully selected and carefully streaked to get pure isolated colonies.
So the experimental plan, all bacterial organisms were obtained from Roosevelt University Microbiology Laboratory, which were initially obtained from the World Science. The positive control used was neomycin sulfate, OK188 and MY09, a test compound that were obtained and synthesized from Dr. Onajole's research laboratory. The materials used, these are the materials we use in the lab. So for the method, so preparation of agar media. Firstly, we sterilize all our materials before we carried out any experiment. So about 2 liters of molar-intin broth was prepared according to manufacturer's instruction. This was sterilized at 121 degrees for 15 minutes using the autoclave. And then we cultured our organisms from the stock into individual TSA, triptych soy agar plate to get our working plate to avoid contamination of stock cultures. Overnight culture preparation. 10 ml of molar-intin broth was dispensed into sterile tubes to use for the overnight culture preparation. We transfer the organism from the working culture plate into the sterile tubes containing 9 ml of broth. Their formation was done aseptically and labeled accordingly. It was allowed to grow in the incubator for 18 to 24 hours at 37 degrees. So for the preparation of the drug stock suspension, 1.024 milligram of each drug was dissolved into 1 ml of DMSO, dimethyl sulfoxide, in each sterile tube, and this made up the drug stock. The working solution for the drug compounds were also prepared from the stock, and this was done by pipetting 1 ml of each stock solution and making up to 10 ml with 9 ml of sterile water. Of all of the tubes were labeled accordingly and kept in different racks to avoid mix-up. Then for the 0.5 McFarland standard preparation, McFarland standard is used for reference to adjust the turbidity of bacterial suspensions so that the number of bacteria will be within a given rate to standardize microbial testing. The absorbance of the organism was checked using the UV spectrophotometer with a 1 centimeter light path of 625 nanometers. This allows for accuracy. 0.5 McFarland standard has an absorbance reading of 0.08 to 0.1. So we had to make sure our organism had such rate for the preparation of the McFarland standard. The organism with highest abundance were diluted with sterile broth to get the correct and accurate values for the McFarland standard. The introduction of solutions into the 96 well plate. Here we used 100 microliters of sterile broth was dispensed into each well using 8-channel pipette. 96 well plates were labeled accordingly to each organism. 100 microliters of each organism was dispensed into the first solution well according to the labels and serially diluted down to the second well. This gave us different concentration of the drug compound and helped us calculate the MIC values for each well, to which my partner Victoria will be explaining the results. The 0.5 McFarland's solution for the organism was also introduced and kept in the equator for 16 to 18 hours at 37 degrees. For the use of the indicator, the INT was prepared according to manufacturer's instruction, and 40 microliters were introduced into each well, which served as an indicator for microbial growth. The white color depicted microbial growth and no drug action. So this is just an image of the workspace in the laboratory. So just as this image shows, the stock drug solution was prepared using DMSO, and the working solution was also prepared. Molanitin belt was also introduced to the wells, of which some were used as a control for the experiment. The bacterial suspension of 0.5 McFarland's solution was lastly introduced and kept in the well. This image shows that we followed the right protocol.
Thank you. Now to results and discussion. This table shows the minimum inhibitory concentration, known as MIC value, for selected organisms used in this experiment. MIC can be determined by culturing organisms in liquid media or on plates of solid growth media. Ours was cultured in a liquid media, sterile broth, just like Dami earlier explained in the methods. MIC is the lowest concentration of a chemical, usually a drug compound, at which biological activity could occur. A lower MIC value indicates that less drug is required for inhibiting the growth of the organism. Therefore, drugs with lower MIC scores are more effective antimicrobial agents. Here are the results gotten from our experiment. OK188 and MY09 showed no activity against all the selected organisms because our starting concentration was 512 microgram per ml, and the INT indicated that it wasn't still enough for biological activity to occur, which made us conclude that the MIC value for both drug compounds against this organism is greater than 512, just as you can see here. Just as you can see here, 512 microgram per ml. Hence, OK188 and MY09 showed no activity, while neomycin, which is a broad spectrum antibiotic, showed a degree of activity and has different MIC values for different organisms. Just as you can see, there are variation in the values. I'll be showing you a pictorial representation as we go back. So here is like the results for the gram-positive bacterial screening. Bacillus megaterium and Enterococcus faecalis was used. If you notice, the Bacillus megaterium, just right here in the 7th well on the neomycin lane, it was still clear. So the drug was still working just before the 8th well, which gives us the MIC value for this plate. The MIC value is 8 microgram per ml, and same occurs in the Enterococcus faecalis, which is 16 microgram per ml. These were also gram-positive organisms, Staphylococcus epidermidis and Staphylococcus aureus. Just like I explained in the gram-positive screening, the same happened in the gram-negative screening, in which this was clear. On the neomycin lane, this was clear. It was all clear, and then it suddenly became pink. The well just before it turned pink is the MIC value, which is 64 microgram per ml. Same happened in Enterobacter aerogenes and its other organism used, Klebsiella pneumoniae and Salmonella typhi. This is our first trial, and these are our results gotten from the experiment. We couldn't continue because of the situation on ground, and further research will be done with time. Triplicate studies will also be done for the experiments for accuracy and replicable results.
So the whole purpose of this experiment is to show the effect of anti-TB compound OK188 and MY09 positive on selected gram-positive and gram-negative organisms using biological screening such as antimicrobial susceptibility testing. According to the results obtained, this anti-TB compound did not show any activity in selected strain of bacteria in the studies. Anyways, triplicate studies are still pending, and further experiments would definitely be done. And for the acknowledgement, we would like to thank Dr. Cordelia Chung for assistance and research discussion during this project. She was really helpful. We also thank the Department of Biological, Physical, and Health Sciences for its generous financial support towards this project. Thank you.
So here is a list of the literature references used in this study. This ends our presentation. Thank you for your time.
Thank you so much.